Everyone, I'm Aneequa and welcome to session 6. In this session, I will teach you how to access different variables and microenvironment parameters using C++. And if you go through these, you would be ready for session 7 next morning, because in next morning, we would be having custom functions that use all the C++, and you need to know how to access these parameters using C++. So right now, I would be telling you how to work with C++ pros, how you can define custom variables in that, how can you query these custom variables using cell definitions, how you can query for microenvironment, boundary conditions, and cell secretion, etc. So this is a basic introduction how you will use C++ in your modeling framework. So first thing, why we need to do that, because C++ gives us all the strength, although we can use the quick builder to actually make all the custom variables and everything, but then you might need to add something later. You might need to use those custom variables in custom functions, and those custom functions would be written in C++. And so the basic where the complexity of a simulation occurs in C++ code. So what files you need to add it? You need to add it mostly custom.cpp. This will be loaded for most cases when you load the template 2D, that would give you custom cpp and custom.h in the custom modules directory. And then you have in the configuration file, you have the PhysiCell setting.xml, and you might need to use it, you might not use it all the time. But custom.cpp and custom.h are the files that you would be mainly using. I can just show you quickly how does it look. So when you use the template 2D, this is what the custom cpp.cpp will look like. It has few functions: create cell types, and then it has set up microenvironment function, set up tissue function, and there are some empty functions for the custom functions, and there is my coloring function here. So this is what your file will look like if you load the template 2D thing. And then there's this custom.h. It is the header file for this. So you will mainly write all this code, which I'll discuss in custom.cpp. Okay, so what I assume before going into this is that we have you already have a model that you have used using Quick Builder, and you can add cell definitions and substrates. So let's assume we have a model where we have 3 substrates: oxygen, glucose, and ECM. We have 3 cell types: wound cell, aerobic bacteria cell, and anaerobic bacteria cell. And then we want to add some custom variables. We didn't edit them in the quick builder, and we want to add them now, for example energy, and there's a parameter alpha that we want to add. This example is taken from one of the project we did using PhysiCell, where we made a simulation of a biofilm, where a biofilm appears on the surface of a wound. So wound gives the glucose to the cells, so anaerobic bacteria can form on it, and the aerobic ones form on top of them, because they actually protect those anaerobic ones from the oxygen, and they get the oxygen from the air, and the anaerobic ones provide ECM for the aerobic ones to get glucose from the wound, because they are on the surface of the wound. So I have linked to the model at the end of the slides. You can go there. So I'm using that because we use that. It is a fully working model, and we can implement it for everything. So first thing is how you can add custom variables. So what are the custom variables? Custom variables are that for each cell, we have extra variables attached to it. You can add as many variables as you want. You can add some parameters, alpha, beta, gamma. You can add some kind of energy parameters. You want to have some extra rates there. Whatever you want, you can add them using custom variables. That would be, each cell will have its own list of custom variables. So how you will add custom variables to model? Either you can use the quick builder but you have already learned how you can use that. You can add custom variables there, or you can use it in the C++ file. And today I'll go through how you go through the C++ file. So if you want to add Using C++ file, where will you add it? You will add it in custom.cpp. That's the file that you will add it. And for example, you want to add to the cell defaults. So you will add cell defaults dot custom data dot add variable. This is a function that you will always call. And this function would take 3 parameters. One is the name of the variable where I am adding energy. Then are the units, and I am adding it dimensionless. You can add whatever the units. And then third one is the value. You will add the value there. So this line of code, this function will add energy data, custom data named energy for each cell. And its value would be initialized with 0.5. Now, you don't want to hard code it like this. You don't want to initialize everything with 0.5. You can always add them to your XML file. And in XML file, you can add it as a parameter, default initial energy, and you can next time whenever you will load this data, you will just change your config file, and this initialization would be taken from that XML file instead of hard coding it here. And then you can also have a another thing. For example, there's an alpha variable I added here. It has no units, and then again I added it using the parameters. So this is how you will add those custom variables to your cells. Okay. The another thing to notice is I am adding them using cell defaults. You can add them to cell defaults, and whoever will copy cell defaults, they will get these custom variables also. But you can also add them directly to the cell, some other cell definition. For example, you have a wound cell, so wound cell dot custom data dot add variables, and you can add extra variables to it. Now the next question is, how will we access these custom variables? So for accessing those custom variables, you will need 2 steps. First, you will need to find the index of that particular custom variable. So you will not access them by name, you will access them by their index in the custom variable list. So first of all, you will find the cell custom data dot find variable index of the energy, and it will result you into some energy, some integer in the form of the index. And once you have that index, then you can access that through pointer, and you can get that on that index you will get the value of that particular custom data variable. So this is how you will access it. For example, you have alpha here, so you will say static in and alpha, so you will get that index, and on that index you will get that custom variable value. And again, we will use we recommend using static because it will be computing only once. Okay. So these were the custom variables. Now, how can we access boundary conditions and microenvironment in the C++ code? Again, where we would be in that our custom.cpp file, and we can be in a custom function, or we can be in the setup microenvironment function, or we can be in any other function, and you want to see what is the value of microenvironment for a particular cell is. So first thing important is to know where our cell is located in the domain. So for that, this function is important. Sorry. This function is important where it will give you the current voxel index. So once you call this function, it will give you the cell is present in this particular voxel. And you can get that, so you know which voxel you are dealing with. Then also microenvironment can find density index of a certain resource. So there can be oxygen, there can be glucose, there can be ECM. So you will find the density index of that resource. So you find, for example, oxygen is at index 0, glucose is at 1, and ECM is at 2. But again, we don't want you to hard code it. We want you to do it like this, access it here and store it in a variable, because if you will add some substrates later, if you will delete some substrate later, they would not remain the same. So we want you to access them inside the code. Then for each cell, we want to see what is the microenvironment value around it, the cell where cell is present, what is the value of oxygen there, what's the value of glucose there, what's the value of ECM there. So there are two ways you can do it. You can either call cell and nearest density vector. So nearest density vector will return the density vector of the size of the substrates. If there are 3 substrates in the environment, it will return a size 3 vector. And if you only want one of them, then you will add that number. And this index will come from here where you have found the uh, density index of that resource, and you will get that resource number, and it will give you only density vector of that particular resource. Similarly, each cell can access the nearest gradient, and gradient of substrate 2 will be accessed using nearest gradient, and in the bracket we are giving it the index. Similarly, we can get all the mesh and mesh voxels. If you want to iterate through all the voxels, you can have a loop, and that loop will go through all the voxels, and you can print their center. And that's this particular way is important if you want to iterate through all the voxels in some function, or you can have different scenarios where you might need that. Okay. Then the other important thing is Dirichlet node, and Dirichlet node is very powerful thing. You can add Dirichlet node anywhere in the domain, and in that Dirichlet node you would have you can actually make a source or sink node there where the value of a substrate would remain constant. So you can add Dirichlet node anywhere in the microenvironment. You can add it before it, or you can want to add it during you would need to add it in C++. So first of all, if you want to add a Dirichlet node, you would need microenvironment.add Dirichlet node function, and it takes 2 parameters, n, and right now I have added BC vector air. So this would be a vector. First I explain this. This vector would be a vector of size number of substrates. So if you have 3 substrates in your microenvironment, this would be a double vector of size 3, and the ordering of those would be 0, 1, 2, whatever is the order of those substrates. And n is the voxel number. So what this line of code would say, it would say on this voxel number, make it a Dirichlet node and set the value of oxygen at a certain point, ECM at a certain point, glucose at a certain point. So it will fix it there. Diffusion will not affect it. It will act as a source or a sink, whatever you want to make it work for that. Okay. I can show you some of the examples here so that you are more comfortable using that. So for example, in our biofilm example, we added you can see here that we added a lot of variables. So this is how we add the variables. Cell defaults, just custom data to add variables. So we added energy rate, use rate, alpha, beta, gamma. So we added all these to the cell. So each cell will have these extra variables with it. Then if you go into the microenvironment section, you can see I have made a Dirichlet node vector air, and this air has oxygen is 20 here, and glucose and ECM is 0 because I want to present air using this Dirichlet node. So for boundary conditions for the air, I made oxygen here 20 and rest of them 0. And so this new vector, whenever if I want to depict air somewhere, I would say dot add Dirichlet node and vector. This would be the voxel. This will be the vector that will give us what is the value of each node there. And another thing is we need to add that Dirichlet activate. We need to activate that Dirichlet node also. So we'll say substrate Dirichlet activation for all of them or for one of them. So if we'll say 0, 1, 2, all of them are true, so it is Dirichlet node for all 3 substrates. But if it is only true for one of them, so it is Dirichlet node for one thing but not for rest of the things. So if some point is we can explain it like this. For example, if a voxel is a Dirichlet node for oxygen but not for glucose and ECM, so glucose and ECM can diffuse into it, but air will not diffuse. Air will always remain constant, and it will act as a source or sink for that particular oxygen. Okay. Next, we have cell secretion, uptake, and export. These we can actually access all the phenotype values from the C++, and we can control them there, but we are just covering cell secretion, uptake, export here in this small session, but you would be able to access everything from there. So again, for secretion, we are secreting certain substrate, and we need to find a substrate index for that. For example, we want it to secrete oxygen. So we would find this oxygen substrate index using microenvironment and find density index of oxygen. So it will give us 0, 1, 2, 3, whatever the integer of this substrate is. Again, always use this instead of hard coding it. Then set uptake rate. So we'll say set defaults dot phenotype dot secretion dot uptake. So in phenotype, in secretion, we have the uptake rate, and uptake rate of what? Uptake rate of the index of that certain substrate. Here it is oxygen, and we will give it some value. Another thing, we are giving this value in the parameters dot doubles. It means this value is given in the config file. The recommended use of it is that we will always give these values in those files, in config files, so that you can easily change them later and you don't need to compile whole code again. Similarly, if you want secretion rate, so you'll get cell defaults at phenotype.secretion rate for the oxygen, and then there's saturation density, which will tell how long will it secrete until it will stop secreting. So if the secretion in the voxel is the value in the voxel is according to saturation density, it will stop secreting, it will not secrete anymore. So this is how you can control the secretion and uptake. And right now I have used oxygen for both of them. So what if it is getting oxygen, uptaking oxygen, then secretion rate and secretion density would be 0. But if it is secreting it, then uptake rate would be 0, and there would be some secretion rate for all of them. Rest 2 of them. Also, when the substrate from the environment is absorbed by the cell, they are internalized substrates, and you can also access them. You can access them using phenotype.molecular.internalized total substrate. Again, we need the index. We need to find the index of that substrate, and then you will give that number here, and it will give us the total internalized substrate. And also we have fraction released at that and fraction transferred when ingested. So these will help you to keep track of those cells. Important thing to notice in all this definition is that if you are in any customized function, if you want to you can want to access the microenvironment, you can access microenvironment at any location. You can make a Dirichlet node there, you can remove a Dirichlet node from there. So that would always be there. The second thing that is important is that we don't want to hard code any index. So if you want to, whenever you want it, you would get a static integer and get index from there, and then use that for rest of the code instead of hard coding it, because it can change and it might give you errors later. Also, another thing is that for XML, you should be making as many parameters available in XML as you can, because then it will be in the configuration file. They will be easy to edit for you. And then the biofilm project I mentioned, this is the code available for it. I want to just go through it quickly for you to see all these things which I mentioned in this, so that you can see that. So here you can see, first of all, whenever I want to access any of these custom variables, I have taken all the variable integers in a static int. So it gives me all the variable indexes, and once the variable indexes are there, not only the variable indexes, but also the microenvironment substrate indexes. So index for oxygen and glucose are also accessed there. And when I have to access them or change them, I will just simply use this syntax to access them, or I can update them. So this is the basic introduction for it. Another thing to notice here is the XML file. So if you see the XML file, there is this user parameters section. So you can always change, you can add extra parameters here, and these parameters will be then accessible in your custom.cpp. For example, in this case, I haven't mentioned here, but I can show you in the So this is the PhysiCell settings for my biofilm project, and you can see all those variables. I have added them in the user parameters section, and then I can access them and CP in custom.cpp, and they would be useful there. So that's it for this section. You would use all these C++ accessing variable functionality for uh, actually coding the custom functions that would be there in the session 7. Thank you.